Hello everybody, welcome to all to my poster presentation session. My name is Emran Ali. I'm a new faculty, Department of Plant Pathology, University of Georgia, Tifton campus. My today's poster presentation title, Statewide Monitoring and Molecular Characterization of Virus Diseases of Georgia's Peanut. Before I go into my results section, I'd like to give you a brief background for this study. As you may all know, virus diseases, mainly thrips transmitted tomato spotted wilt virus, have a great potential economic impact on peanut production in Georgia. To minimize this virus disease impact, it is really very important to have an up-to-date survey record in Georgia. So far, small-scale surveys may have been conducted in Georgia for virus diseases, but not yet a statewide survey for molecular characterization of virus isolate in Georgia. So moreover, some other tospoviruses that can easily infect peanut, like groundnut ringspot virus, GRSV, tomato chlorotic spot virus, TCSV, those viruses are already recorded in southern Florida, but not recorded here in Georgia. All this scenario has put a tremendous pressure to the peanut industry in Georgia. To address this issue, with the funding help from National Peanut Board, we have conducted a statewide survey to develop a monitoring program for virus diseases in Georgia's peanut. Further, we also characterize TSWV isolate based on their virulence. The survey has conducted May 2019 through August 2019, so total 18 counties in Georgia are surveyed, a sample per county 20. So we have tested for 3 different tospoviruses, TSWV, GRSV, and TCSV. As you see the figure 2, here's the Georgia county map. The blue colored area, we have collected our sample. All those samples are processed and characterized at Plant Molecular Diagnostic Lab, University of Georgia, Tifton campus. For testing this method, we use 3 different techniques, mostly RT PCR. To compare this method, we also conducted ImmunoStrip and DAS-ELISA testing method. For RT PCR, we have targeted N gene, a nucleocapsid protein gene, and a NSM gene, non-structural protein gene. For the ImmunoStrip and DAS-ELISA, we have followed the manufacturer instruction to detect this virus. In figure 3, you see an example of testing method result for 3 different methods. In the top panel, you see the 2 different markers for TSWV, TSWV-N, and TSWV-NSM gene. If you see these specifics band size that can represent this sample are positive for TSWV or not. For the ImmunoStrip test, if you see the 2 representative band that told you that can represent this sample are positive for TSWV, and if you see just single band that represent this sample are negative for TSWV. For the DAS-ELISA method, as you can see, if you see the dark yellow color that represent the highly positive for TSWV. If you see less colored yellow colors, well, that represent the also positive for TSWV, but the virus titer is low. If you see no color changes, that means this sample are negative for TSWV. Now I'd like to jump in our result section. So first we did the compare among these 3 methods, and based on our result, we see RT PCR is the most sensitive assay for the detection of TSWV. However, ImmunoStrip, as you know, is very convenient for the rapid on-site detection for TSWV. DAS-ELISA is a very lengthy process, and it's a less expensive method, and it's very suitable for large number of sample detection. Now I turn into our survey result. Based on our survey, we have found total 92.3% of our sample are infected with tomato spotted wilt virus. The rest of 7.6% sample are turned out negative for TSWV. So then, to see the other tospovirus present on this sample or not, we have tested with other markers like GRSV and TCSV markers, but we found negative for them as well. So this negative sample might not be even pathogenic. It's just simply a nutritional problem, or maybe infected with some untested pathogen. Throughout the survey, we have also noticed that, as you see the figure 1, we have different types of symptom-based sample we receive. We receive severe sample, we receive mild symptomatic sample, and we also receive looks like very healthy, maybe just only 1 or 2 necrotic lesion present there. That type of sample we also received. So that can also tell us that the symptoms for this TSWV are so variable depending upon the age, weather, and variety, and host plant. In further, we also compare some of our TSWV isolate in compare with NCBI submitted sequence for GRGS isolate, and our result indicate that the both N gene and NSM gene, their sequence identity is ranging from 97 to 99% in both nucleotide and amino acid level. Very few nucleotide or amino acid are different among isolate. Due to the limited extent of sequence variability within a small number of TSWV isolate we have tested, no significant relationship were identified among population. So I can conclude our summary for this study. From this survey, we have found only TSWV tospovirus present in Georgia. By comparing all 3 methods, this study suggests that ImmunoStrip is suitable for on-site detection. However, if you have a well-equipped laboratory, RT PCR is more sensitive and reliable method for pathogen detection and quantification. The slight variation of sequencing variation of N and NSM gene among TSWV isolate might have a potential role for the fluctuation of symptom severity, but we could not prove that theory in this study. Further, a detailed study would be needed to establish the relationship between those nucleotide or amino acid changes with disease severity of TSWV. Finally, I'd like to thank all participating UGA, county extension agent, and peanut grower to provide a sample for this study. Finally, thank you so much for your patient hearing.